our intro, what that's all about. Next slide. So I think I've got about 10-ish minutes of background that I'll try to get through and then get out of the way so that Marlene can present really the meat of what we're here to discuss with you. But first, for background, the N1 Collaborative is an independent coalition. It's a nonprofit that we set up a few years ago. And our mission statement is that we're committed to developing repeatable, rigorous, and responsible pathways for individualized medicine. And this is really a genetic therapies group. Our goals here are to try to draft best scientific and clinical practices for individualized genetic medicine, to share data about successes and failures, and hopefully through these types of activities to increase access and to make it even and democratized across the world. Our website is shown there. Next slide. This group that we started has some 700 members that participate in various activities. We have work groups within the N1C on various topics that have 65 volunteers. And we have 9 core operational team members that are helping coordinate all of these activities. Next slide. And here are pictures of some of the folks on our team. This is the core operations group. Our names are way too small, and there are, I think, a bunch of slides of pictures, but key points, key faces here to point out are Hugh Hempel, who is our recently appointed executive director. There's Steph Leonard, who's on the call today, and one of the, and together Steph with Nicole are the 2 full-time employees of the N1 Collaborative. And Margaret Meserve is a genetic counselor in my group. I'm here. Next slide. The board of directors, this group was co-founded by myself and Julia Vitarello. And we have members from the Global Genes Organization, Nicole Boyce, as among our board of directors, Anamika Artsmarus, who's been a pioneer in genetically targeted therapy using antisense oligonucleotides, is on our board. Winston Yan works as our volunteer president. And here's our executive director again, and Holm Grassner runs, among other things, the Solve-RD network in Europe. So he's maybe well known to some of you from the gene discovery standpoint. Next slide. SAB members, well, let's just go through this and try to get to some more explanation for why we exist. Next slide after this, and this is us here on the call. The reason that the N1C was created was it really began out of a project that my group led beginning some 7 years ago, where we met a patient who was in the midst of a diagnostic odyssey. This was a young 6-year-old girl who had progressive neurologic decline and a provisional diagnosis of a neurodegenerative disorder called Batten disease. It was provisional because not all of the genetic mutations underlying that condition could be identified, but whole genome sequencing in her showed that she had an unusual causative variant. There was a deep intronic variant that altered the splicing pattern of the CLN7 gene. The recognition of this, the completion of this diagnosis allowed us to develop a customized therapy for her that could alter splicing patterns and restore normal gene expression. And in the next several months after realizing that that opportunity existed, we were able to make good on it in the next slides, showing that we could restore normal gene expression and do sufficient animal safety studies and regulatory filings to actually launch a clinical trial for her. And this work in the next slide was published now 5 years ago as an example for how the certain genetic diagnoses have therapeutic implications and could be used to actually develop medicines that might actually be conceivable to be developed within a clinically relevant time span for that patient in whom you found the original finding. Next slide. That work, accompanied by two other examples of rapidly developed individualized oligonucleotide treatments. One advanced in Columbia by Neil Schneider, and another one advanced at the University of Massachusetts with Bob Brown and Jonathan Watts, formed the basis of this, this concept that some genetic diagnoses have exceedingly important therapeutic implications that, that could allow them to be turned into actionable treatments. And that was really the motivation for starting this N of 1 collaborative, to try and incubate this idea, to figure out what the rules of the game are, to figure out under what scientific and ethical parameters such work could be considered with very serious conditions that are debilitating or fatal, for monogenic conditions, and for conditions that are otherwise too rare to be the subject of commercial development. These are the types of considerations that formed the basis for FDA guidance that was released in 2021 that allowed this type of work to proceed with a standard regulatory approach, and is the starting point for how the N of 1 Collaborative was formed, to try to think about these guidances, to improve on them, and to also fill in the gaps for the many, many things that were not laid out in the guidance themselves. Next slide. I'm actually going to skip through. There are a couple of slides here with additional examples, but I want to shortcut through to get to Marlene. And I'll say that one of the big gaps left in the guidance is how do you identify, what are the genetic cases? How do we recognize the genetic variants for which these types of individualized approaches might be appropriate? That's left completely unspecified in the guidance because it's really not a regulatory question. It's a genetics and scientific question. But I will point out that that's the subject of our coming to you today. I will point out that in the last 5 years, since we first published that paper, there have been a total of 24 patients that we are aware of who have received therapies under this type of individualized guidance to date. That's 24 as of October this year. And I suspect that a lot of them were just actually dosed in the last 2 months. And there are many more coming in the next upcoming 6 months. So it's a field that's not entirely esoteric. It's not hundreds of patients, but it's dozens of patients. So this is something which we think we may be seeing more and more of, which is why I think it's relevant for discussion today. Next slide. Just some of the other players in the space. So there's many global initiatives now that are working to try to develop these kinds of these programs in the Netherlands, the Dutch Center for RNA Therapeutics, where Marlene is from. There's a European network called the One Mutation, One Medicine Network, including investigators at these countries highlighted in blue that we partner with to develop European regulatory standards for this type of approach. There's groups in Japan, Takanori Yokota, who are working with Japanese regulators to create new guidance in Japan for it. And in the UK, there's an initiative announced last year that I'm a part of. And there's another slide about an Australian group here too. And the point of this is just to say that there's a lot of activity in this space and opportunities for us to kind of get a handle on it and figure out how to establish some harmonized approaches towards it between these different groups. St. Jude's as well. So I will try to now get out of the way and hand things over to Marlene to take it from here.
Thank you, Tim. Very nice. I have to try to kind of adjust my synchronizing with your slides. Yeah, so the N of 1 Collaborative, as Tim said, is not themselves developing the treatments, but we're trying to provide resources and guidance and advice as much as we can to everybody who is trying to develop N of 1 treatments. And currently, of course, the focus is on antisense oligonucleotides, but we are not going to restrict ourselves to that forever. And we are trying to organize ourselves currently in different working groups. So there's multiple working groups everybody can join if they're interested in and participate. And one of the working groups I would like to introduce to you today is the patient identification working group, because we think this is the one that's most important to the work that the ClinGen Consortium is doing, and where we also are hoping if we can partner up, collaborate, and work together in the future. I also have way too tiny names on my slides, but I'm trying to give you a little bit on an overview. So in our patient identification working group, we really have many different people involved. So we have clinicians, we have pediatricians, neurologists, neonatologists. We have ethicists that are involved in our group, but also researchers from different backgrounds. So we have bioinformaticians, biologists, geneticists, many different people from across the globe that have been trying together to find ways how we can select and identify patients and cases that are most eligible for our treatments, are most likely to benefit, and also make sure that we prioritize the cases that are most urgent. So this is the group we are working with. There's people joining and going all the time, but this is basically the core group we've been working with in the past 2 years, and it's been very fantastic. And I also very much appreciate that there's many young researchers joining us that are really giving a lot of contribution and input to our work. So when we look into which cases are most likely eligible for the currently antisense oligonucleotide treatments, we are always focusing on 3 pillars. So one thing we are looking into most is the genetic variant. Is this a variant that's a good candidate for any of the antisense oligonucleotide approaches? We also have to look into the disease. Is it a disease that is treatable? And of course, currently we are mainly looking into diseases that are progressive, and also diseases that are manifesting in tissues that are treatable, so that where we can deliver our compounds, and that is mainly the brain, but also the liver, and nowadays you can look into the muscle and the kidney as well. But mainly at the moment we're still focusing on neurological disorders, and we also have to see, are there phenotypes that we can measure, and things that we can actually have an outcome where we can look if there's a clinical benefit. So while biomarkers are great, we really want to have clinical outcome measures where we can see there's maybe a slowdown, a halt of the disease, maybe the loss of vision is not progressing, something in this direction, or the amount of seizures or frequency duration is going down. And the last thing we have to look into is the patient themselves, right? So every disease you can have in different stages. So patients are progressing throughout their lifetime, and we have to ensure that while the treatment development can take from 1 to 4 years, depending on how successful, how fast we are, and if we're running into any obstacles, we have to ensure that the patient is still in a treatable status the moment the compounds are ready. So these are the 3 pillars we are focusing on, and we actually have within our patient identification working group subgroups that are specializing in these 3 areas. And what we want to talk about today are the variants, because this is where we really think we can work with you together. And also, let's say it's the easiest that we pick, because often, or in many times, it is a clear no for a variant. When it comes to yes, it gets a bit more complicated. And also, this is where we currently have done the most work. So we've recently, or the last 1.5 years, we've been working on what we now call the N1C variant guidelines. So the acronym is Variant Assessment Towards Eligibility for Antisense Oligonucleotide Treatments. And 2 weeks ago, we put out the preprint, and you can now access all the information we have put together. So the guidelines are about 60 pages, and I will just quickly introduce you to them, how we developed them, and what is inside of the guidelines. So this was a multi-site effort. It did not only include our patient identification working group, so that was the core group that was initially preparing the outline, but we later took in lots of assessors, so people that tested those guidelines from across the world. And I think that was very beautiful, because we got to meet lots of people that did not have any background in ASO technologies or in variant assessment for these type of cases. And so that was great for us. And the guidelines were developed with us having already looked into about 1,500 cases totally. So within our working group, that's the amount of cases we have been assessing in the past 2 years. And from what we got, the knowledge and the expertise, we tried to develop guidelines to help other people learn how to assess these cases and for themselves decide if they have good candidates. This is also an overview of all the people that took part, so all the assessors that participated in our studies. And as you can see, they were from across different countries, different institutions. And again, we had lots of different backgrounds. So bioinformaticians, but again, clinicians, clinical fellows, established professors, and also master students and PhD candidates. And all of them were beforehand naive to what we were doing, and they were sent those variants and then had to they were sent the guidelines and then had to assess some cases. So how did we do that? Within our working group, we did a first draft. And from that on, we got feedback from different people within the working group, and we revised it. And then we had a first pilot where we sent it out to a smaller group of assessors with a few test variants just to ask them, do you understand this? Can you help? Can you interpret the variants when you're reading this? Can you make an assessment? Which of those test variants would you think are eligible for an antisense oligonucleotide therapy? And from the feedback we got, we then kind of drafted new, like we revised and drafted new. And one thing we really looked into was not only the feedback themselves, but also did the assessments go well? So I didn't care if someone was giving the right answer. And we asked everybody when they assessed a case to explain how they come up with the assessment and why did they give a certain assessment to a case. And based on how they were reasoning, we tried to then revise our work to make sure that the way they get to their results is the correct way. And we did that a second round, again, drafted a new version, got feedback from the core group, revised again, piloted a second round. In this round, we had 12 variants that we sent out. And then we did a third round where we did it again at that point with 15 variants. And we sent it, I think, in the last round to about 20 different people. And again, got feedback and asked them to assess the cases, asked them to explain how they come up with the assessments of the variants. And based on that, we finalized our work. And as I said, pre-printed it 2 weeks ago and also submitted it to a journal. We are looking into different types of ASO strategies. So the guidelines are not just for one ASO strategy, they're covering multiple ASO strategies. That's why they are so long. And it's also that not you as an assessor would have to read the whole guidelines, but you can go into the specific section that is relevant for your variant. And the strategies we're looking into are splice correction, canonical exon skipping, transcript knockdown, and what we call upregulation from the wild type. This has also been published at a different term. It's called TANGO, targeted augmentation of nuclear gene output. And that's a term that was given by Stoke Therapeutics. And I'll just quickly show you what these strategies mean, that you understand what types of variants we can assess with this. So when we talk about splice correction, we often mean variants that are either deep intronic or deep exonic that are either activating or creating cryptic splice sites. And then they lead to, for example, if they are deep intronic, to an inclusion of part of the intron, which will lead to a shift in the reading frame, and thus no protein produced or nonsense mediated decay. And you can block these with an antisense oligonucleotides. And what is beautiful with these variants, you normally can restore canonical splicing and then get the full protein. That's one way. Another way that you can do is canonical exon skipping. So these are often missense variant, truncating frameshift variants, leading to an early truncation. And if that exon fulfills certain criteria, you can remove this exon from the transcript with an antisense oligonucleotide. You will get a truncated or a shortened version of the transcript, but you can still get a partially functioning protein. The other thing, what you can do if you have a gain of function variant or duplication of a gene, you can go for a transcript knockdown. So you can use so-called gapmer antisense oligonucleotides, and they recruit an enzyme, RNase H, which then degrades your transcripts, and you can thus downregulate protein levels. And the last thing is that upregulation from the wild type. So if you have heterozygous variants that are causing haploinsufficiency, you can try to use the wild type transcript that is still intact and upregulate. And here, for example, you can do poison exon skipping and thus produce more canonical transcript. You can downregulate the natural occurring antisense transcripts. By downregulating those, you can also increase the the protein product from the wild type, or you can modify and regulate the UTRs. You can do that with the 5' as well as the 3' UTR. So there are multiple ways. So all of these are very different strategies, but it is fantastic because for every variant that you have that might require a different strategy, you can now go into these guidelines and look up which of the strategies are most likely eligible or suitable for your variant, and if your variant is a good candidate. And the guidelines follow certain steps that you can just take. You can really, it's a step-by-step guideline. You can go through it and follow them. And first, of course, we have to make sure that the variant follows HDVS description to make sure you're looking at the right transcript, at the right variant. You will have to look into what is the pattern of inheritance. That's not always known, but ideally, we have this information. We will have to know what is the pathomechanism of the variant, and we are also aware that this is not always known. You can deduct some things, but if you're really unsure, we recommend people not to assess these variants. We look into splicing effects, and we've gotten very strict with that, because nowadays we say, if there's no evidence against splicing, check for splicing, just because we've seen that many missense variants and many of the variants that are often overlooked are actually causing some splicing effects. And then in the next step, you can check which of the ASO strategies are most likely suitable for your variant. And then you can go into the section of the guideline and read up how to assess your case. Yes, so this is how it then looks. You have an overview table. It gives you the inheritance patterns and the type of pathomechanisms. And then once you know this, you can look up which of the strategies is useful for you, and then you can go into the certain strategy, into the chapter of the guidelines, and read through that section, and then assess if your candidate is eligible for treatment. And we've been trying to identify classification terms, and this has been a discussion within our team on multiple occasions, because we know that there's never a clear yes, and there's most likely also never going to be a clear no. There are a few cases where we would say it is a no for an ASO strategy, but there are debates about this. So we've now been trying to classify them across a spectrum, and we did 4 classification terms, but we are most likely going to refine them over time, because we already know that a few of these variants fall in between. So we have at the moment not eligible category, and an unlikely eligible category, likely eligible, and eligible. And eligible are cases where we really have evidence that this is a good candidate. So that can be, of course, that there's already a treatment available. For exon skipping, it can be that we know that there's patients walking around that are missing this exon, so they have an exon deletion, but they're healthy candidates. So these are cases where we say, well, they're definitely eligible. Likely eligible cases are everything that within the guidelines kind of follows the guidelines, but there's not yet functional or proven evidence in a patient that this is going to work. Unlikely eligible cases are based on the guidelines, based on the molecular biology we think is not going to be suitable. That might be, for example,
you can go ahead.
Marlene is on mute.
Oops, Marlene, somehow you ended up on mute.
I don't know. Yeah, all good. Yeah, so for an unlikely eligible case, so if you have an exon where there's a functional domain, so if you would skip that exon, you would lose that domain, we would currently call it unlikely eligible, unless, of course, you have some functional data that shows that it's okay to lose this domain. And then everything that currently cannot be categorized, or where we don't have enough information, where the pathomechanism is not known, where maybe the inheritance patterns are not really clear, this is what we at the moment would fall into the unable to assess category, and you would have to gain more evidence, more data to then be able to assess the variant. And we assume that over time we will, of course, revise our guidelines, and then variants might have to be reclassified in the next year, in the next few years. So this is something that we know is fluid and dynamic. This is not fixed, but we had to come up with the first classification term at this point. So this is basically how it looks like. I'm just showing you one of those tables. This is for the knockdown approaches. So you have the eligible, likely eligible, unlikely eligible, and not eligible categories. And then these tables list all the criteria. So you have a whole text that guides you through it, but then you have for every ASO strategy an overview table where you quickly can look up the criteria for each of the classification terms. And we did not end there. So we wanted to give people more guidance, more help, more training material when they're now reading those guidelines, when they're practicing. We are aware that's not something you read once and then you just know. It will take practice. It will take some time. So we have about 30 practice variants that you can test your knowledge on. So you go through the guidelines and then you can follow the practice variants. And there are answer keys that we provide in a separate file that you can then look up, which explains how we assess those variants. We also provided training videos. So there's 12 instructional videos that basically cover all the types of variants and all the types of classifications that there are. And they will guide you through it. So you can pick some examples and just use those training videos. All the tools and all the material we are using in our guidelines is stuff that is publicly available. So databases, tools that everybody can use. So it's accessible to everyone and there's no restrictions. So no licensed tools. You are allowed to use them, of course, but we wanted to make this as accessible as possible. And I think David, who did these videos, did a fantastic job by guiding people through it. And these are short videos, 3 to 6 minutes, and then you can follow how he has been doing these assessments. And then what we also did, because we know this is labor intensive and this takes some time and you can get lost in the guidelines, we built a variant eligibility calculator. So instead of reading through everything, you can also use the calculator if you have enough information on your variant and you can click yourself through it. So if you know what is the variant, if the inheritance pattern, the variant type, you can go through it and you can fill in everything in the calculator, and the calculator will give you the results of your assessment. So then, for example, for exon skipping, there's multiple criteria and you click which ones apply to the exon that you want to skip, and then it will tell you if this is an exon that can be skipped or not. And also what you can do, you can log your variant assessments with your variant. You can type it in the day and time. And if you go through it and answer the questions in the calculator, it will log your answers. And at the end, you can also download your assessment that will help you keep track of everything. But also if you have to reassess a variant in the future, you will see what has changed, how your answers changed, or if the assessments has changed because the guidelines have been updated in the meantime. Yeah, as I said, this can now be pre-printed at medRxiv, so you can download it with all the supplements. The calculator is online. You can find it on our website, on the N1C website, as well as the videos there on YouTube. You can also go through them from our website, and then you can use it. And since this is still under review, we are happy if anybody of you has already some feedback or some improvements or ideas. We are very happy to take this in. And our idea is that we now publish this paper once, but that the guidelines itself will stay up to date continuously. So they will be updated on our website regularly. And then you can find the newest version of the guidelines always online. What are we going to do next? So first of all, we want to train some people to use them, because the idea was always not that only 5 people in the world are able to do these assessments. We want Margaret is laughing because you know how much work it is. So we want to do this, or we want everybody to be able to do this. And this is how the guidelines were written. So we really tried to do it as understandable as possible, as simple as possible, so that people can follow this, practice themselves, and then do it on their own cohorts. We have multiple people in different countries that are testing those guidelines on their cohorts right now, seeing how many of their cases are eligible. We will be doing workshops in the upcoming months to train people if they're interested to learn directly from us. And of course, always feel free to contact us. But our dream or our vision is that right now, this is, of course, still a lot of work. You still have to train, and it will still take some time to assess a case. So ideally what we want to do, and we are working already on the background to automate this whole process, that all the information that you now have to fetch yourself, an algorithm will fetch for you and basically go through those questions in the calculator automatically and then give you the results of the assessment. So in the future, we can hopefully do this faster and more accurately without any human errors. You still will have to look into it. I don't think we will get rid of that, but it will go faster. And you can do that in bulk. with that, I would like to thank you for inviting us or for allowing us to speak today. I would like to thank, of course, anyone and everyone at the N1C, our board, scientific advisory board, our operations team, the whole working group that has been putting a lot of work into this, and of course, all the volunteer assessors and co-authors that we randomly grabbed at conferences. I don't know how many people I just approached said, "Oh, you liked my talk. Don't you want to become an assessor for our new project?" And then, of course, also all the patients and families that have been submitting all their variants over all these years and allowed us now to build this resource for everyone. And with that, I think we will end here. And the idea was that we answer some questions, and I think there were already quite a lot in the chat. So thanks, Margaret and Tim, for already answering them. And then, of course, we would like to open the discussion how we can work together, how we can partner up, and how we can just share our expertise and resources and hopefully bring more of these treatments to patients across the globe. Thank you, everyone.
Great. Thank you all very much for the talk. If anyone has questions, please go ahead and use your reaction, raise your hand. Otherwise, we can go through some of the chat questions just so you're able to answer them out loud for anyone who is listening. I think one of them we had here was just talking about your ASO modality, and is that currently what you're using only? Do you plan to expand? You want to talk about that?
Tim, maybe you are, I think, the best to answer this question.
Sure. So I think in some domains, like variant interpretation, we are focusing on ASOs because practically speaking, that's where there is some work to be done. Our ambitions are that a lot of the other work products of this group, thinking about what are appropriate clinical trial designs to support very, very small patient-sized clinical studies, what are the appropriate ethical and regulatory frameworks for approaching these studies, et cetera, et cetera. We hope that those will be generalizable because we are imagining that when CRISPR really kind of gets headwinds and is being rolled out to address individual mutations, that all of that work will be necessary. So we position ourselves as really trying to think through the clinical trial readiness for a future when these other modalities are on board. But when it comes to variant identification, we're focusing on something more practical, which is how do you get some consensus as to whether or not a particular genetic variant might be eligible for consideration in one of these research studies. Does that make sense?
Yeah, great. I
I
think
mean, we can talk
another,
about base editing, but it wouldn't be a practical value at the moment because there's no, one, it'd be too easy because the rules for what's base editable are kind of defined biochemically. But two, there's no practical regulatory path for individualized base editing at the moment. The moment that there is, then we should come up with some guidance.
I think a really another interesting question in the chat that came through is during your presentation, you were able to show many of the different areas of the world that are covered by collaborators and people working, but one of them was just asking whether or not you are exclusively US, Europe, Australia, and whether you have representation from other countries, if you wanted to speak to that for a minute.
You know, as a group, we just want to put out, plant a flag and say that if you're working in this space, we would like to speak with you. And so any places of the map that aren't covered, and there are a lot of them, are places where we just simply haven't had folks self-identify yet. And so we're not restricted, partially because we also don't make any promises since we're not the ones developing the drugs, right? We're really here to try to serve as a place where folks can talk through difficult issues. That makes it easier to invite anybody, since we're not making promises about being able to deliver a drug in their country, so to speak.
Great. That's really nice on that clarity. And of course, it sounds like you want to engage anyone to use your tools too. So hopefully getting more engagement internationally will be really great. One of the questions I thought was interesting was around talking about duplications and autism, but in the answer, it was also very interesting, if you want to speak more to it, Tim and others, about the considerations that you have to have, right, around which particular targets you have based on FDA guidance and your therapies.
Yeah, so this is really interesting. So Marlene and David and the patient identification group have done a really nice job, I think, in laying out, what are the molecular genetic criteria, as well as other criteria too, that could create an opening for a research project to be spawned to develop a therapeutic. But there are lots of other things that come into it that have nothing. They're not molecular genetics, and one of those is just the prevalence of the disease. And current N of 1 guidance, the spirit of the guidance was to allow for work to be done in diseases that are considered orphan, meaning they're below some threshold for attracting commercial investment. But exactly what that definition is not clear. The FDA guidance was very conservative in saying that we anticipate this may be most cases where this guidance may apply are cases where there may be only one or two cases in the world. But even that's not completely defined. Is it one or two cases per year? Is it one or two cases ever? Is it one or two cases available to enroll right now? There's a lot of gray area here. And so when the question about duplications came up in the chat, that's another really good example. Dup15q is rare. It's not attracting a lot of commercial investment that I'm aware of. But it's more prevalent than certainly one or two, and its prevalence is sufficient that you could recruit a healthy number of individuals in a trial. And in that case, it's important scientifically to know that you could have an ASO strategy to treat a condition like that. But practically, the regulations may not allow for that to occur under the N of 1 guidance. So maybe it's big enough that you could get company investment, and maybe you would have to use a more traditional clinical trial approach to bring that treatment to bear. We kind of stay outside of that a little bit. We're not, that's the subject of other working groups in the N1C about proper regulatory parameters and not so much about patient identification per se.
Great. I think that was really helpful information, Tim, to be able to tell us. Now, there's a really nice question, I guess, just if anyone is interested, that came from Melissa Ritono. It's just, could patients or parents of the patients contact you directly to be considered to be evaluated? And I know you said yes, but wanted to know, do you have an email you can pop in the chat or provide for everyone? Just so, again, anyone here has that, or again, do they go to your website? Is there a contact that if you can give some information for the group that might be useful?
Yeah, that's a good question. And I'm not entirely sure what to say here. So folks email Marlene all the time. Folks email Margaret in our group all the time. And folks do email Stephanie all the time. And by pointing to Marlene, I'm pointing to her as a representative of the Dutch Center for RNA Therapeutics, a center which has put out their flag and said, hey, we are in the business of evaluating cases in Europe and trying to work along these paths. And emailing Margaret and me, it's putting us in the context of me and our flag planted at Boston Children's. In the context of Stephanie, who's as one of the full-time employees of the N1C, we're actually, as a group, we're trying to be like ClinGen and not like a diagnostic reference lab. So we are trying to help coordinate the activities of people who are working in this space, and not be necessarily a hub where patients go to get their answers directly. That's just a matter of bandwidth. But of course, like everything's fluid right now. We're a baby organization, unlike ClinGen. We're trying to learn from ClinGen and how you've set yourselves up so nicely. And so in these stages, yeah, email Stephanie and we'll work it out. Because we're just trying to answer questions and get information out there.
Oh, great. I see Ryan.
Hi, thanks, Tim. That was great. Were you guys engaged with the FDA in their development of the ASO guidance? That's question one. And then question two, have you had any conversations about something similar for AAV?
So technically, N1C as an organization didn't exist when the FDA guidance was released, technically, right? But I will say that the FDA guidance was based largely off of meetings that we had. We had a critical path innovation meeting guidance with the a critical path innovation meeting with the FDA in July of 2019 that was, I think, the stimulus for the guidance that emerged. A lot of the ideas that we discussed in that meeting were basically what came out in the guidance. And then the guidance also has benefited from lots of public comment from other parties, including some of us who were organized before under a different title before the N of One Collaborative existed. A lot of the core members of the N of One Collaborative came from a rare disease task force convened by the Oligotherapeutic Society. That was myself, Art Krieg, Anamika Arts-Marbus, who is one of Marlene's mentors, and John Watts and Keith Gagnon. So there was input into that, and there's continuing input from lots of other folks. And Lorem, which has a lot of folks from Ionis Pharmaceuticals, has also contributed a lot to the guidance, especially in the manufacturing side of things, because some of their CMC people, their chemists, have volunteered their time for the NLorem Foundation and given the FDA extra suggestions for how to responsibly manufacture these in a safe way.
And for these other emerging modalities, is there any movement from the FDA to come up with a similar guidance?
There's a lot of pressure on them to do this. Interestingly, in a lot of these other modalities, like CRISPR, aren't under their purview. So ASOs fall under the purview of the Center for Drugs, and it's the Center for Drugs which released this guidance. It's been hailed by many as being amazing and transformative, and we're really happy about it. Let's let 24 patients be treated. It's also been criticized by some as being unnecessarily still too restrictive. Interestingly, the other modalities that are the next generation modalities that are coming, like CRISPR and direct mRNA therapies, are under the jurisdiction of the Center for Biologics. And there, Peter Marks, on his own accord, is going out and actually talking about very much N of one style guidances. It's not codified in the guidance, but he's out there talking about it. And in his slide decks, he's really pushing for even more, I think, creative, flexible frameworks for platform strategies to use all of these therapies as flexibly as they can. There's not a question about this, but can I volunteer? Can I volunteer a question?
Absolutely.
So one question that Marlene and Margaret and Steph and I had coming in here was, well, what do we actually want? How do we want to engage with all of you? And the answers, there's lots of different answers, but one of the questions in our minds was, we've explicitly tried to model some of the activities of developing these variant guidelines along the way that Heidi, you set up the variant interpretation guidelines with the ACMG, and trying to think, well, how do we standardize this? Just very, very broadly, like how do you standardize the approach, right? And we'd love to kind of continue to talk with you and learn from you about the lessons that you learned and how can we use that to improve our guidelines. How do we make them as rigorous as possible? How do we make them as quantitative as possible? So that's one line of activity. It did occur to us, well, should we, and are there groups within ClinGen that would be especially useful for us to speak with? And I think that depending on the subtree of the variant classification that you go through, if you're talking about a splicing amenable mutation, then talking to your splicing group would be really useful. If you're talking about criteria for identifying a toxic gain of function mutation that deserves knockdown with, say, an ASO, then maybe talking to some of your gene curation groups might be appropriate. So that was one area that we were thinking of, trying to ask for your advice. It did occur to us, well, at some point, should this be incorporated into, should this type of interpretation be something that is reported in genetic testing reports? And I think that's a really interesting question. It's also kind of a loaded question because the moment you begin doing that, then it sets up expectations and it puts a lot of pressure on a system that is still very much in its infancy, and this is still highly experimental work. So I say that, but then pull back from it a little bit, and it would love to get your thoughts on that. However, there is one specific scenario where I wonder whether or not earlier integration is called for. And that's that people in our community are creating mutation-specific drugs for a lot of different orphan conditions that currently have no treatments. And they aren't always registered in clinicaltrials.gov, and some of them some of them actually are being delivered as compassionate use under named patient programs in Europe, and there's no structure for organizing them. And so we have a discoverability problem because we have, for instance, right now running a trial in ataxia telangiectasia that targets a very specific recurrent mutation in ATM that's found in 10% of the Turkish population. And there are many folks who've migrated to places outside of Turkey. And we have a discoverability problem because if someone encounters that variant right now, they may not know that we're running a trial, and they may not read the paper that we published describing the trial. So we wonder whether or not ClinVar some ClinVar-like mechanism might be used for variants for which an investigational protocol is active and available, whether ClinVar might be a way to connect people to those existing research opportunities.
Great. I know you had a lot of questions, Tim, there, and we'll definitely address them. I do want to see there's a hand raise from iPhone, and then Heidi will go to as well.
Oh, sorry. This is Sharon.
Oh,
I didn't realize that I hadn't logged in, so it just said iPhone. Hi, thanks very much for the talk. And maybe you said this at the very beginning, but I do think something between asking labs to report on it, which I think would actually be very difficult to get them to do, is I do wonder whether any of our VCEPs might want to include, when they classify a variant, looking at how hard would it be for them to include in the variant summary or do it in some structured way with you, whether that variant meets your eligibility. Because that takes it away from the diagnostic labs that I don't think would want to get into it, but might be something that VCEPs might be interested in.
So at the level of an expert curation panel,
Yeah. So, I mean, the ClinGen one, right? Obviously, there are lots of outside groups. But.
Yeah, I would. Can you hear me? By the way, I'm on
Yes.
convoluted thing in the airport, but I agree with Sharon that probably asking the labs to explicitly define suitability for N of 1 is maybe not the way to go. Combination of just sort of jurisdiction of what the labs should be doing in terms of patient care, but also just resources and stuff. But the ability to point towards resources that can assist in that as sort of a standard direction, like labs could easily put some sort of resources there to say, to kind of point to a resource or an approach or a method or something. I do think there's lots of ways that we could interact between your group and ClinGen. We have a sequence variant interpretation working group that then has a bunch of subgroups of computational splicing. We're about to form a functional work group. And so I think there's a lot of ways to interact. There's even the potential to have your group actually, if you were interested, be a subgroup of the ClinGen SBI system or some sort of partnership between the N of 1 Collaborative and ClinGen. It was a joint venture in essence, but then interfaced with our various groups that might bring additional expertise to weigh in on the various topics that you brought up. And then someone else wrote in the channel chat, the ability for us to prioritize with our expert panels which genes and variants we curate because they are potential to inform treatment. That's already something that we all encourage all of our groups to do. So having some sort of more formalized way to to have a feed through of what is in that category, I think would be great as well.
Great. I wanted to see we have 5 minutes left. If there are any other questions? Okay, well, I want to thank you all for joining us today to go over your